This video will be talking about the logic of natural selection and the genetic components that Darwin wove into his theory, the genetic observations in particular that he wove into his theory in order to figure out how natural selection worked. So natural selection, as I've discussed in the previous videos, can be broken down into ecological and genetic components. And when it comes to genetics, there were two key observations related to genetics or hereditary factors that Darwin leveraged to come up with a theory of natural selection. These genetics observations relate to how phenotypic variation occurs in a population and how that phenotypic variation is heritable. Darwin didn't know about DNA or even chromosomes or genes at the time, but he did know about phenotypic variation and how organisms often resembled their parents. So genetics observation 1, phenotypic variation occurs. It's a relatively straightforward observation, but it's a very important one. All populations have variability, and individuals in a population are not all identical. Marr can't really say it any better than that, but I'll tell you the way that Ernst Marr said it. No two individuals are exactly the same. Rather, every population displays enormous variability. Throughout these genetic examples, I'll be using human skin pigmentation as an example of this. This is a trait that varies tremendously between individuals, it varies within populations, and it varies across populations. There's an interesting example of this. It was an art project, I believe it's still standing in Pittsburgh downtown at the August Wilson Center, and it's an artist that has done installations around the world. She takes photographs of individuals and then matches their skin tone to a background, or rather matches a background to their skin tone. One of the goals is to show the variability of human skin color and how simple words like black and brown, white don't really encompass all the variation and variability of human skin pigmentation. So within Africa, we often talk about Africans having dark pigmentation or being black, but if you actually think about it or take some observations, or in this particular study took highly quantitative observations using a light meter and to measure what's called the melanin index, you can see that within a single population in Africa, in this case the Botswana San, there is a wide amount of variation. So on the x-axis here is what's called the melanin index. They used a photometric device that allowed them to measure how much light was absorbed by the skin, and they converted this to how much melanin was in the skin, and the size of this peak indicates the most common, so most individuals fell along here along the scale, but there's a wide variability within this one population, within this one ethnic group in Botswana, wide variation in how pigmented their skins were. If you go to another population, in this case the population from Ethiopia, they were all much darker pigmented. The Botswana San, their median is perhaps around 70. This group in Ethiopia, their median is about 125. But each one of these groups we can see has a large amount of variation within the population, and then there's also wide variation between them. So both ethnic groups, populations within Africa, but dramatically different degrees of skin pigmentation. Now, if you look across Africa, in this particular study, they looked throughout Eastern and Southern Africa, and they found a whole wide array of different distributions. All of them are approximately normal. Every population has variation. They've ordered them in this particular figure from the Botswana San, who are relatively lightly pigmented. As you increase through these different groups, they've ordered them. They become increasingly darker. So throughout the continent, there is wide variation between populations, and within each population, there's wide variation. For example, the Botswana San, relatively lightly pigmented overall, but the darker pigmented individuals would actually fit in within this Ethiopian group, the most highly pigmented group. I'll do this one over here, it's better. So this individual is the most highly pigmented, darkly pigmented individual of the Botswana group. They fall within the left-hand side. They would be relatively lightly pigmented in this Ethiopian group, but they would not be out of place. So even this most lightly pigmented group is so variable that it can have individuals that are as pigmented as individuals of a darkly pigmented group. So lots of variation within traits in human beings and in many, many organisms. We often take it for granted in humans because we vary so much in skin pigmentation, in height, in hair color, in hair morphology, so many different traits, but all organisms have this amount of variability. Birds vary in how big their beaks are. Lizards vary in how quickly they can run. Deer vary in how much muscle mass they have. All organisms vary. So a second observation is that much of this variation is heritable. Obviously, many physical traits can vary with nutrition, with food, how tall humans grow, how large our bodies are, how much muscle mass we have. That is heavily influenced by our environment and by our diet, but it's also influenced by our genetics. So a lot of variation is due to heritable factors. Again, Darwin didn't know what these were, but he could definitely see the pattern that offspring often are similar to their parents, and offspring then pass those traits on to their offspring. So grandchildren often bear similarity to their grandparents. Great-grandchildren bear similarities to their great-grandchildren. My daughter, we can see traits that she possesses that are similar to her great-grandfather, and that also occur in some of her second cousins. So we can see how that trait gets passed down through the generations. Marr can't really say it much better, but I'll give him a chance since this is ultimately his framework. So much variation is heritable. So to continue on with skin pigmentation, there are a large number of genes that have been associated with variation in skin pigmentation. Here's a list of 22 from a recent publication. And many of these genes have multiple alleles. So depending on which alleles of which genes you have in combination will determine your exact skin pigmentation. So the fact that there's so many genes and each gene has different alleles, the effects add up to determine exactly how darkly or lightly colored an individual is. So here's data from a single paper where, again, they measured the melanin index. They took a light meter, they put it on people's skin, and they determined approximately how much melanin they had in their skin. And overall, the population had this kind of mean distribution. And then they genotyped some individuals on the upper and lower end. So they sampled some individuals on the upper end of the distribution to determine their genotype for a single gene, and then they determined those on the lower end, the ones in the gray, in the middle. They must not have had enough money to sample these individuals, so they just focused on the extremes of the distribution, those with the highest melanin index, those with the lowest melanin index. They were looking at a gene this is a single gene with two different alleles, the big A allele and the big G allele. Individuals that were big A tended to be, now there are many genes, this is just one of 20 or more genes that impact skin pigmentation. So being AA, big A won't make you definitely be light-skinned, and being GG will make you definitely dark-skinned, but it can influence it. This is a gene that happens to have a fairly large effect. So individuals that were big A, for this gene tended to be on the lower end of the distribution. There was variation here. All of this tan color here are individuals that were big A. The other variation that these individuals all have the same genotype. They don't all have the same melanin index because other genetic factors, all the other genes that are involved in it, and environmental factors. Some individuals that were on the spectrum were heterozygotes, big A, big G. So some heterozygotes at this gene for these two alleles were on the spectrum. Then individuals that were big G tended to be darker pigmented. The most highly pigmented individual in the population was big G. The second, looks like the third and fourth most individuals were all big G, big G. But there's more variation here. Many individuals could be on the higher end of the spectrum if they were heterozygotes, and even a few big A individuals were actually very high. So these are individuals that even though they have this big A gene, or the big A genotype for this particular gene, even though most people that are big A are lightly pigmented, these maybe 3 or 4 individuals were darkly pigmented. They must have alleles for other genes that are driving their overall pigmentation pattern. So any given gene, any given allele does not determine exactly how highly pigmented an individual is, but this happens to be a gene that at least this allele being homozygous big A does tend to make these individuals on the low end of the pigmentation scale for this particular population. So again, this is just a single gene, has 2 alleles, big A and big G, and you can stop it and review the text here for if you would like to review, so you can pause the video. So there are many genes that contribute to skin pigmentation, and these have been even looked at the molecular level by looking exactly at the number of micrograms of melanin that are in cells. So here's the MATP gene. It has 2 alleles, big F and big L, and you can see homozygous F individuals have the lowest on the melanin index in this. This is a cell-based assay, so this wasn't looking at individuals. These are cell lines that have these different genotypes. So they're trying to get at kind of a mechanistic cellular biology basis and actually directly quantifying the amount of melanin. So higher value here means more melanin, more darkly pigmented. FF individuals are lightly pigmented. FL individuals are medium. LL are most darkly pigmented. And there's other genes that were examined in this study. There's the NCKX gene and the R129 gene. Each of these genes happens to have, or they investigated 2 different alleles of the gene. So for NCKX5, there was homozygous T, heterozygous TA, and homozygous AA. And again, this homozygote has low melanin, this homozygous has high melanin. The R129, 2 alleles for the gene, CC has low melanin, TT has high. Now, all cells have all 3 of these genes, and they created cells that were either homozygous recessive for all of the loci or homozygous dominant for all the other ones. So here on the left, this is a cell where you can see the dark splotches are the melanocyte. There's only really one spot right here. So this is a cell that is FF, TT, and CC. So altogether, this produces a very lightly pigmented cell. This is a cell that is homozygous dominant, LL, AA, TT. So homozygous dominant for these 3 genes makes a very darkly pigmented cell. So we understand the process of pigmentation and the variability caused by different genes and allele at the molecular level in this case. So these are the 2 genetics observations that Darwin marshaled, and these are going to come together and be combined with the ecological observations to start to infer the process of evolution, and I'll discuss that in the next video.